This is Dr. Patrick Hu, president of SITC, and we are so happy today to have Dr. Carl June with us. This is a man who needs absolutely no introduction. He is currently the director of translational research at the Abramson Cancer Center at the University of Pennsylvania. He's made so many contributions in his lifetime, both to basic science, translational science, clinical science, has saved many lives by his clinical trials. And he is really unparalleled in how he learns from each patient that he treats as well. So, Carl, thanks so much for joining us today.
Thank you so much, Patrick, and for that very generous introduction. It's really a pleasure to be here with you at SITC.
Great. Let's start at the beginning of your career. A lot of people may not know, but you're a graduate of the Naval Academy in Annapolis. Tell us how you got there and how you got interested in immunology, starting from the Naval Academy.
Yeah, well, that wasn't always the plan. I was actually admitted to Stanford and when I was in high school, and then I got a low draft number because the Vietnam War was ongoing. And so rather than go in the infantry, I went into the Naval Academy, and so you could go in as an officer. And then when I graduated, the Vietnam War was over. So the Navy then decided and allowed me to go to medical school. And our biggest military issue then was the Cold War with Russia, which is rather poignant where we are today. But back then in the early 80s, the issue was that with the Cold War was nuclear confrontations. And we had submarines that we wanted to have to monitor Russia. And to do that, we needed to have a bone marrow transplant program within the military because of limited nuclear casualties, as we've learned, for instance, in Fukushima. And so the Navy trained a small number of physicians in bone marrow transplant. And so in 1983, I learned how to do that, and then got very interested in graft versus host disease and what was a really awful problem back in the early 80s with the early days of bone marrow transplants.
Yeah, so you did a lot of transplantation research at really one of the meccas of transplantation, the Hutch. And you also have had some real diverse experience with the World Health Organization and with malaria. Can you tell us a little bit about that?
Yeah, well, so the Navy is, by law, congressional cancer research goes through the National Cancer Institute as a direct line item. And the Navy does two kinds of research, which are infectious diseases and combat casualty care. And at the time, I had about a 10-year obligation to stay in the Navy after the training. So I worked initially in malaria and then in HIV. So that's how, for me, it actually turned out to be beneficial. I learned a lot about lentiviral vectors, which came from HIV. And then, later on, we used that in CARs. And at the Fred Hutch, I have to say, it was really some of the first translational research they did in bone marrow transplants. And, I was part of a program there where they did a trial comparing allogeneic bone marrow transplant to syngeneic. They actually had enough people where the siblings could be monozygotic twins, identical twins. And we went into that experiment thinking it was better to be a twin donor if you had leukemia than it would be to be a usual brother-sister homozygous matched for chromosome 6, but not an identical twin. And it turned out, all counterintuitively, that the people who had the best leukemia-free survival were not the twins, but rather the siblings, that had mismatches, and we now know triggered graft versus leukemia reactions.
Yeah, so that was your first experience really with what is now T cell therapy and
Yeah.
specificity. So, before we get to the CAR Ts, where you're really widely known, you have made really important contributions to co-stimulation and basic science of immunology. You want to talk a little about that?
You know, I began, got interested in how T cells grow because we saw graft versus host disease, in spite of, at that time, the most targeted immunosuppression in general that we could give for graft versus host disease was corticosteroids, and then I was involved in a trial with cyclosporin, which came out in about 1985, and it turned out we found we could grow steroid resistant T cells and cyclosporin resistant T cells if we co-stimulated them. And so in the lab I was at the Fred Hutchinson Cancer Center with several others now, Claudio Anicetti, Paul Martin, and John Hansen. I showed as a postdoc that CD28 co-stimulation made the T cells resistant to immunosuppression. And so we studied for about 10 years how to grow T cells and what controlled the switch between being sensitive to immunosuppression and being resistant. And that led to some of the later work with CAR T cell signaling domains.
Wow, so it really all came together from the basic immunology into the clinic. You've been one of the most influential translational immunotherapists in the cancer space. What inspired you to start to focus on cancer?
Well, we saw in leukemia that an allogeneic marrow transplant was better than an autologous or syngeneic. And that was completely unexpected then. So then, I began to work initially with HIV T cell transfer because the Navy wouldn't allow me to work with cancer. And so we worked on growing T cells with gene transfer. We made the first CAR against HIV. It was called CD4 zeta. And we did that, in patients who had HIV before there were potent antiretroviral drugs. And so we learned a lot about the principles of growing T cells, human T cells in large scale. But my basic training in medical oncology had been leukemia. And when I left the Navy, I went to the University of Pennsylvania in 1999. I began working with a team there on leukemia patients and using the same principles of growing T cells that we had used in HIV, but for cancer and for CAR T cells in leukemia patients.
Tell us about the CAR T work and how you got started on it and why you focused on lymphoma initially. I mean, that has been so successful, saved a lot of lives with that work. What inspired you to do that?
Yeah, so that, this was something that, the more junior investigators need to understand. There was initially a lot of work, both by biotech and academia, trying to make gene transfer therapy work. And then there were a few side effects, lethal, that were unexpected in the late 1990s, and that biotech left all the research up. So there was only a few academic centers working on gene transfer technology, and you were one of them. You saw what Steve Rosenberg did with TIL cells and trying in the early days of gene transfer. And there was, Stan Riddell, Fred Hutch, and Phil Greenberg working, and Michelle Sadelain, and just a few, literally a handful of investigators that were still trying to do gene transfer and T cell immunotherapy. And the field really showed that, leukemia, especially the B cell tumors, are not very susceptible to transplant. So it became that we knew rituximab was, really one of the first targeted therapies for lymphoma, targeted CD20. Michelle Sadelain, Werner Brenchen showed that CD19 was actually a better target. It was expressed earlier as a lineage antigen. And so, several of the groups, the NCI, my group at Penn, and the Rosenberg group, as well as Michelle Sadelain, made CARs targeting CD19 as a demonstration of the CAR. And, the pharmaceutical industry had ignored that as a target. They were comfortable with CD20, rituximab. and it turned out CD19 is actually probably better, and lots of work on that since then. So our initial 3 groups, targeted CD19, and part of that was that we knew that congenital immunodeficiency with B cells was tolerable. Kids could be born without B cells, and then as long as they got gamma globulin replacement therapy, they were okay. So it was not like some other immunodeficiencies where, for instance, T cells are absent, and which proves lethal. So we knew an acquired B cell aplasia could be manageable if we could also target leukemia through CD19.
Well, it must be so fulfilling that your work has gone on to get FDA approval and has saved so many lives. There's a recent publication that you had just showing the long-term effects that really a lot of these patients with hematologic malignancies getting the CAR Ts that you designed are being cured. How does that make you feel?
Yeah, well, I mean, it's a rare event where, actually the CAR T cells in humans have worked better than they did in mice. I mean, one of the dirty secrets we all have as immunologists is human T cells are more robust than mouse T cells. And so it actually worked better. The first patient that Steve Rosenberg treated who had a lymphoma with a CD19 CAR has had very long-term, I think a decade-long survival. And what we just published in Nature was that 2 of the first 3 patients that we treated with leukemia, are cancer-free a decade later, and they actually are still engrafted with the CAR T cells. And so that was completely unexpected. We started that experiment in 2010, and we were using a mouse hybridoma, for the binder to CD19 called FMC63. And we thought that the humans, with these leukemia, even if we eradicated their leukemia, they would eradicate the CARs, because they were, expressing a mouse protein that they weren't tolerant to. And what we found is they did not do that. They have long-term expression of their autologous T cells that express the mouse anti-CD19. And those cells, stay functional in the patients now for a decade, with a single infusion. So it was really the first time a living drug could be shown to be installed into a patient that way with gene modification and have, a target and tumor specific effect.
Well, it's fascinating. And it also demonstrates what I've been so impressed with, and that's how you analyze the therapeutics in the patients for how long the T cells last, where the insertion sites are. I mean, really very exciting. You've learned so much from that paper where you looked at the insertion site of the retroviral vector and you came up with TET. And even when you've seen toxicities, what the epitopes are and the motifs. I mean, I've just been really impressed with how you've analyzed carefully and deeply scientifically the patients to get additional information. So, and that's not easy to do because you have to get the samples, you have to process the samples, you have to analyze the samples. A lot of times you don't have grant funding for that. So how have you done such great deep analysis of your patients once you get treated and where you learn so much?
Well, that's a key that, unfortunately many pharmaceutical trials really only look at the bottom line and don't invest much in sample preservation. And so we were fortunate that these were academic trials and we could save cells and, for later analysis. And that's just been invaluable as you just recounted. So the fact that, these cells are gene modified is unlike, if you treat someone with a cancer chemotherapeutic, it goes, their body's metabolized, you can't really study very easily resistance mechanisms. But with these gene modified cells, you can study their fate, their state. And we learned with the very second patient we treated who had acute leukemia, she relapsed with a CD19 negative leukemia, so target loss. And then we and others have now found that in lymphoma, target loss isn't the primary mechanism resistant. It's either direct lymphoma resistance to T cell killing or trafficking issues in the tumor microenvironment. So the resistance mechanisms are different with different CD19 targeted CAR cells, and having samples to study that has been really critical. And I think that's a lesson. The clinical trialists need to be teamed up in a matrix fashion, with basic scientists. So, we found in the 10th patient we treated with CLL that he had a very late regression of tumor and a tumor lysis syndrome. It was actually 50 days after his infusion. And what we found was those were clonal proliferation of T cells, but it had, as you mentioned, Patrick, a loss of function due to insertional mutagenesis with the CAR in the TET2 gene. And we didn't know why that might have happened. We've now found it recurrently. So it's not an accidental stochastic insertion. That those cells survive longer if they have insertion into that locus. And it's since been found that, with age, clonal hematopoiesis or CHIP occurs in elderly patients, and where they have the loss of function of TET2 in their hematopoietic stem cells. So human stem cells and T cells have more stem-like behavior if they have loss of function of TET2. And so that was, we were able to show that with, bedside back to bench studies because the cells are genetically marked.
That was such a beautiful paper and a beautiful story. And also, if those cells survive longer, are you trying to do that on purpose now?
Yeah, that is clearly. So we found out, initially that this happens by studying the exceptional patients. And as you know, that's often a good way to understand either resistance or exceptional responses. And so now Joe Faretta here at the University of Pennsylvania, and he's working with Wei Ming Kong and also with Matt Porteous at Stanford on doing this intentionally, using, Cas9 to make targeted insertions into the TET2 locus. And that looks very promising, where it could be done intentionally rather than by virtue of the near random insertion through a retroviral vector.
Yeah, I've also been impressed that you've been a rapid adopter of new technologies. When you were doing lentiviral, lentiviruses come from HIV. People were pretty wigged out about it at first, and you were really the one. We were all using retroviral vectors, which we, for the most part, use a lot still from mice. But you started out with lenti, and you were one of the ones that really led the lenti revolution throughout all of the regulatory things that you had to do to get a virus that's related to HIV for clinical use. And so that you're one of the first to get CRISPR in patients accomplished. So you've really been a rapid adopter of new technologies. Where do you see this headed, and where do you see the future lie for cell therapy?
Yeah, well, I think that's a great question. So there's low-hanging fruit, I think, when you have intersection of, several fields. So in the case of immunotherapy, it's been the realization of neoantigens. We didn't know why TILs worked. What you and the surgical branch have now shown, that was completely unexpected, the mechanism. And then the fact that you could use, so there's mutagenesis to make neoantigens. And now with vectors, we can insert basically targeted neoantigen into T cells. T cell receptors are CARs. And so those are add-ins with viral vectors, and there's many ways to do that, and even now non-viral ways, Sleeping Beauty or Piggyback, or and so that we're really fortunate that T cells can be, modified that way. I think now that what's really going to be wonderful is this genome engineering and synthetic biology, that the ability to make T cells synthetically with multiplex editing has this huge potential. And I think we'll see, and that in the end will be something that, giving checkpoint therapies works in many cancers, but there's many where it doesn't work. And I think synthetically engineered cells are going to be able to break through that glass ceiling.
Well, that's exciting. We're trying to get immune therapy and cell therapy to work now in the solid cancers, which is the vast majority of cancers that people die of. What do you see for the future of that, and how do you think this will be with TILs versus CARs, which is the antibody and the signaling chain versus getting the natural TCR in, or even a fusion? Michelle just had a paper that looks at a fusion of a TCR and a CAR. What do you think it's all going to look like in the future? Someone's going to get cancer, go to the doctor 10, 15 years from now. What do you think the treatment they're going to get consists of?
Yeah, well, I think the dream we all have is, I mean, first of all, there'll be precision therapies. And we know, as you know, I mean, all cancer, they may have phenocopy each other in many ways with the pathways that we've learned about, but the specifics of each mutation are unfortunately not the same. So we're going to need precise understanding of cancer biology of each particular tumor. I mean, just now the whole KRAS thing, it looks like we maybe can target KRAS, but there's many alleles of KRAS, and there's different solutions for the different alleles, and similarly for, for instance, BRAF and so on. So I think target, we're going to have both understanding precise of the tumor but of the host immune system. And in some cases, the host immune system's already been either ravaged by therapy or by the tumor itself and so suppressed. I mean, many leukemia patients present with opportunistic infections. I mean, their immune system is not working. And so I think we're going to have allogeneic off-the-shelf type approaches for those, if the immune system is not working. But if it's working, in solid tumors, there's a major issue with specificity. And now that can be engineered into cells, whether it's a TCR or a CAR. I think so. Specificity is a huge issue, where there's not so many mutations as there is, for instance, in melanoma, where there's often many neoantigens there. So in tumors with low mutational burden, gene transfer, can fix that. But then there's an issue just of having enough effector cells to deal with the tumor burden, and then do they traffic into the tumor? And there's big barriers in many different tumors that glioblastoma, whether the blood-brain barrier will be intact or not, or whether pancreatic cancer, there's fibrosing scars around it, and how do you get penetration of drugs, antibodies, or T cells, for instance, into a tumor that has a stromal wall around it. So, but I think all that can be engineered, and it's going to involve combinations, not just of T cells, but it'll be NK cells, macrophages, and oncolytic viruses I think have a huge potential and to be combined with cellular therapy. So I'm excited that, we'll see all that. The problem is how long it takes to do these trials, and the turnover time. If we can make that more rapid, I think the ultimate ability to take and translate what we know in basic science now about how cancers are caused, how they can be fixed, can be clinical therapies. But right now it's a very long time to get these things into clinical trials and into the practice of medicine.
That's a very important point. I remember reading about Bell Labs and how they developed the transistor, and it was what they really did is the process. They made their idea translate so they could figure out if their idea was good in a week or not to get to the next idea. And so what you're saying is that we really have to compress that time when we have an idea and a thought to get that into patients much quicker. That's a very important point. And it also brings up the point about a lot of people look at cell therapy. They say, well, it takes so much labor. It's personalized. It takes all this gene manipulation. It's too expensive. It's hard to scale. What's your thoughts about that, about the scalability of cell therapy to make it a common therapeutic in the future?
Yeah, so I think it's, people didn't think that you could vaccinate 1 million people with RNA, even 3 years ago. So they learned how to scale an RNA vaccine because there's a lot of effort, for that. And I think that technology now, is scalable. I think we're going to have both, as I mentioned, autologous cells and then off the shelf allogeneic. They may be iPSC developed and derived or from healthy donors and cord blood. But I think so. In the case of allo cells that are not patient derived, I mean, they'll be there and be used on demand. But I also think the FDA and physicians will become more comfortable with genetic modification of cells, and it will become more like blood bank modeling, where T cells and stem cells could be harvested locally, and at point of service be modified with turnkey systems. And so I think engineers are going to solve that problem of scaling this. And we now have a trial open at the University of Pennsylvania using where the manufacturing of autologous CD19 CAR cells is 3 days. So, I mean, right now the label for the Novartis Kymriah CD19 CARs is 22 days vein to vein. And so now, the manufacturing is much shorter. And I think it's going to end up even becoming shorter than 3 days. And so and the fact if it could be done without the complex logistics, which it is now, of, for the commercial cars, the patients are apheresed, it's shipped to a central manufacturing center, and then returned back. Very complex manufacturing, but I think if it happens at where the patient's located, then it's scalable in a much better way, and it could be a one-time therapy.
And the other topic you brought up is testing, getting the testing to happen quicker. We may not be able to do 1 week like Bell Labs did for the transistor, but what do we need to do as a society? And what can we do? SITC does a lot of conferences where we bring together the government, academic, industry. What could we do to try to move trials along faster so we can test an idea quicker?
Well, I think SITC is actually doing a lot of that. I learned from a bone marrow transplant experience where I think a lot of it is really relevant to the scaling out of cell and gene therapy. So bone marrow transplants, in the early 80s were really only done at the Fred Hutch and at Hopkins, where there were, the large centers involved to do that. And then it over 2 decades spread throughout the country and out through worldwide and into not just at central medical centers. And so one major issue is workforce training. And SITC has a major effort in that, both in junior investigators and the older physicians now who are trained 30 years ago have no idea what TILs are. They don't know, if they're a solid tumor oncologist, they've never done apheresis, for instance, and made cell therapies like bone marrow transplanters have. So the workforce needs to be trained, and whether they're senior physicians or new junior physicians, they're learning that. And so assisting in the workforce training is a major issue to decrease this barrier from, small pilot trials to being implemented in the practice of medicine.
Wonderful. And speaking of training, you have trained and mentored so many people. I mean, we've all learned so much from you, but you have personally mentored so many people. I think I saw a tweet today. I think it was by Bruce Levine. He said, really appreciative that he was your postdoc 30 years ago, and he's gone on to be a big leader in the field. So what's that meant to you to have so many people now leading programs that came from your lab?
Well, it was really a small cadre, as I mentioned, because the biotech was not involved in this initially, just a very few academic centers. And now we've had this seeding worldwide of, from the centers that, where you were initially at the NCI and Memorial Sloan Kettering and Penn and the Fred Hutch. And it's gratifying and tremendously to see, those people, and then they train more people to see, this adopted in a general therapy. So I think that's one of the most alluring parts of an academic career, is the fact that you can see people, it's international science, and we can get past political barriers and have we all have now children that were in our lab and now are carrying things on the flags forward elsewhere. And that's tremendously rewarding to see. And I just hope that other people get to experience that with, and as we come out of COVID, to be able to, see shoulder to shoulder again what we've lost in the last couple of years.
There's really a great next generation coming along that are interested in sciences. What advice would you give to them if they're just starting in the field of immunotherapy and oncology?
Yeah, well, I mentioned, I think, right now, for instance, this area of synthetic biology with the immune system. So understanding genome engineering with the immune system has huge potentials. Certainly, a skill set I don't have is AI and large scale bioinformatics. So people can't learn all of those things, but if they work with teams, then they can take on big problems. And so that's one thing, is learning how to work in a matrix environment. And the other I always say is this thing that Yogi Berra, a catcher on the Yankees, used to say, if you come to a fork in the road, take it. And that happened to me where several times, I mean, I had trained as an oncologist, and then I had to go into HIV research because the Navy, for instance, was not allowed to do cancer research. And that turned out to be really beneficial. So that was an unplanned career fork. And I've had several of those that way, of going from basic science then mostly into translational trials and so on. And so you need to somehow get out of your comfort zone. And unless something pushes you to do it, it's hard to do that. So in my case, I trained in bone marrow transplant, but then could only get grants in HIV. So I had to learn how to become a virologist. But that then ended up having big benefits. And I think the same thing right now for, people learning gene editing, gene manipulation, bioinformatics, understanding cancer biology. There's just tremendous promise there. It's such an exciting time to enter the field.
Well, that's great advice. You've been such an important member of SITC through the years. You've received the highest honors from SITC, most recently inducted in the Academy of Immuno-Oncology. Through SITC, you're going to help to honor Zela Geshar next year in our annual meeting in Boston. We really appreciate you doing that. What's it been? What's the meaning for you to have been a member of SITC for so long?
Well, it's similar also to bone marrow transplant. I mean, it was a club initially of just a few fanatics, of which you were part of, and then and there was no industry with it, so we struggled year to year. When I was on the board at SITC initially, I mean, it was literally, could we pay the bills to keep the lights on? I was astonished at last year's meeting. So the growth now is exponential. It's really important because of this workforce training I mentioned. That the society grows and keeps bringing this, because it's a new area that's not covered elsewhere by, as far as I'm concerned, by ASCO. Really SITC's been the one society that has long-term said, what can we do for immunotherapy and solid cancers? Which I think we all agree is the number one issue now. And SITC has really the platform to go after that central issue.
Well, we really appreciate what you've done for SITC as an incredibly valuable member of our society. You're also a ton of fun to hang out with, and you have a lot of interesting activities that you do. Why don't you tell us what you do for fun?
Well, my most recent obsession was during the COVID was, we couldn't do the usual thing I like to do, which is extreme sporting events, because they were all closed down. So I spent a year and a half doing what's called Everesting, and this started in New Zealand, but it's in one bicycle ride climbing the height of Mount Everest, which is 29,000 feet. And I did that last August in Maui on Haleakala. I went up and down Haleakala till I got 29,000 feet, and And so that's the most recent thing. It keeps me it keeps me sane, I have to say.
That's so impressive, Carl. I've gone down Haleakala on a bicycle. Can you go down? But I can't imagine going up and down until I've got the height of that. That's extremely impressive. Hey, I know you've been around the world, but you've lived in Philadelphia for a number of years now. So tell us about the town. If you were going to sell Philadelphia, would it be the Eagles or what would it be? The cheesesteaks? What would you sell about Philadelphia?
Well, Philadelphia, actually, because I lived in the Bethesda area, as you did for a long time with NIH, um, and the housing price is much lower than in Philadelphia than it is in DC or in New York. And then, for my own, there's wonderful bicycle and hiking in the Philadelphia area. We have Fairmount Park, which is similar to Rock Creek Park in Washington, DC. It's this huge area that people don't know about Philadelphia, where you can hike and bike, mountain bike, and so it's a nice livable area that, and now it's really become, what people probably don't know is that Philadelphia and that eastern Pennsylvania was really home in Pittsburgh to the polio vaccine. And that was done initially with eggs, so there's a big industry here in the pharmaceutical area for vaccine technology. And so that's something a lot of people don't know. And now it's becoming a cell engineering area that Bruce Levine has called Silicon Valley. So the Delaware Valley now has many, many different spinouts doing cell and gene therapy.
Wow, I didn't realize that it had that background in the vaccine industry. Knew about the cheesesteaks, though.
Yeah, there's lots of cheesesteaks. Yeah.
That's great. Carl, anything else you'd like to bring up today?
Well, I really I can't wait to have this with Sally Gesser next year. So for those of you who don't know, I met him several times at the Weizmann Institute, and he made a CAR molecule, and he didn't do it to try to cure cancer. He did it on a basic science experiment. He wanted to find out, could you make a T cell that wasn't MHC restricted? As the immunologists know, you need to have HLA identity between T cells and its target. So he grafted an antibody domain onto a T cell receptor and found out that he could do that. And then later, after showing that in model systems, he showed that you could kill tumor cells that way in mice in an MHC unrestricted fashion. So that's a huge lesson. That was done in the late 1980s, and took decades to be translated into humans, but it was basic science. And now we're seeing the same thing. For instance, I mentioned genome engineering and how it has become close to cell therapies of cancer. So that's a really exciting principle, and I'm hoping that the new the future of SITC is to accelerate that pathway
of taking the basic science to the clinic. Because you're right, that initial antigen specificity was trinitrophenyl. Really had nothing to do with cancer. Against hapten.
Yeah.
Well, Carl, I'm just so impressed. Every time I talk to you, I learn so much. You've taught the whole society. You've taught the world so much, science, how to translate, and also how to be as a human being. You're one of the most humble guys that I know, and just you're very impressive person. I can't say what an honor it is and how thrilled we are that you spent time with us today.
Well, thank you for doing this, and I really appreciate the opportunity to work with SITC. Thank you.